Hi, my name is Grant Kramer, and I am a professor emeritus at the University of Nevada, Reno. Today we will be doing my sixth lecture.
Plant Physiology, Lecture 6, Phloem Transport.
As you recall, we are covering a variety of different transport processes that are responsible for the plant to be able to capture the resources that it needs. First, we discussed water transport and the mechanisms and how they did that. Then we covered ion transport and how they captured mineral nutrients from the soil. And today we will be covering phloem transport. After the plant has created photosynthates from photosynthesis, how it captures those sugars and transports them to other parts of the plant where it's needed. In this video, we will be emphasizing the transport of sugars in angiosperms, the flowering plants, and only briefly discussing some of the issues that have arisen with the study of gymnosperms, the very limited amount of studies in gymnosperms. We will be talking about the patterns and pathways of transport of the sugars in the phloem. We'll be talking about the composition and the materials inside the phloem and the rates of transport. And in the end, we will discuss a model of how sugar transport occurs in plants. To fully understand the mechanisms of sugar transport and the model that will be presented at the end of this video, you must understand the mechanisms for water transport and ion transport and osmosis. This was covered in earlier videos, so I encourage you to go back and review them if you're not familiar with them.
So there are two long-distance transport pathways in a plant, the phloem and the xylem. And as we previously discussed, the xylem is primarily responsible for the transport of water. In the phloem, it can also transport water, but it also transports sugars and other important compounds that are necessary to be distributed to all the different parts of the plant. So let's look at the phloem anatomy and a stem here as a cross-section. And what we see are 4 cell types that are involved in the phloem tissue. Phloem is a tissue, and it's made up of individual cells that include fibers, sieve elements, companion cells, and parenchyma cells. We'll talk about these in more detail in a moment. But if you look on the right here, we can see the cross-section of a vascular bundle. And on the outer edges of that vascular bundle are the bundle sheath cells, some of which are sclerified as sclerenchyma or fiber cells. And in the interior, we can see the primary phloem towards the outer edge of the stem and the xylem on the inner side of the stem. And this is typical of vascular bundles. The phloem cells are living cells. The xylem cells are dead cells. The cells are staining red because the dye that is used stains lignin, which is in the thickened walls of cells that are mature, and certain cell types only. The cells that are involved in the transport of sugars are called sieve cells. And here we see some figures drawn of an individual sieve cell on the left-hand side and several sieve cells forming a sieve tube on the right-hand side. The sieve cell on the left is typical in an angiosperm, which has at the end of it a sieve plate with large pores in it. And then on the side of the cell are lateral areas with pores that are much smaller that allow transport in and out of the cell. On the right-hand side, we can see multiple sieve cells aligned to form a sieve tube. And notice there are several components of the sieve tube cells. There are components such as plastids inside of it. There's also P proteins, and there's large spaces in between the cell that allow the movement by mass flow of sugars through the cell and into the next cell through these large pores in the sieve plate. There is a plasma membrane around the cell, and the P proteins that we are talking about here have a special function. They line the sieve cells along the edge, and they're utilized when a sieve cell is damaged, such as a rabbit eating the end of a stem or a leaf, chewing it off. These P proteins will quickly move to the sieve plate and plug up the pores, causing callus to form and plugging off the sieve cells or the sieve tubes and preventing them from leaking out all the sugars out of the plant. Note, there are no nuclei in the sieve cells. As these cells mature, some of the normal cell components are removed. Along with the sieve tubes, there is another cell type that's very important for the functioning of these sieve tubes, and that is called the companion cell. In fact, they're both derived from the same mother cell and split into two. And the companion cell is adjacent to the sieve cell, and it does have chloroplasts and nuclei and mitochondria. And notice there are these many branched plasmodesmatal connections between the companion cell and the sieve cells. So it is thought that the companion cells are providing energy and nuclei control, regulatory controls to the sieve cells when it cannot produce them themselves. So they go hand in hand with each other. In addition to the companion cells, there are other cell types such as parenchyma cells, which act to store and then release food molecules into the sieve tubes. And there are fibers and sclerids, which are used for protection or strengthening of the tissue. And in some cell types, like in lettuce, there are things called lacticifers, which are latex-containing cells. But these are not common to all plants. And in some cases, as we'll talk about later when we talk about different photosynthetic processes, such as C3 photosynthesis and C4 photosynthesis, there are some cell types called bundle sheaths that surround a vascular bundle and are particularly important in C4 photosynthesis. In trees that have secondary growth, the phloem is in the inner layer of the bark. And this is why if you strip off the bark of a tree, you effectively kill it because you're removing its ability to transport sugars from one part of the plant to the next. So let's look at an electron micrograph of a cross-section of sieve cells and companion cells. And here in this figure, we can see a couple of sieve tube elements where the arrows are pointed, and you can see a companion cell adjacent to it. And you can see that the companion cell has a much thicker layer of cytoplasm with many organelles in it, whereas the sieve tube elements are relatively empty with a very thin layer of cytoplasm, which allows for the transport of sugars easily through these sieve tubes. These few organelles and materials in the cytoplasm of the sieve tube elements are along the periphery in order to reduce the resistance to mass flow. In this next figure, we can see a longitudinal section of the sieve elements and adjacent companion cells. And you can see at the sieve plate, where two sieve elements are joined together to form a sieve tube, that there are pores that are formed. And these pores are open. They are not occluded. In early studies of these types of images, the cells were damaged and the P proteins would plug the pores, and it was thought that these pores were plugged. But now with better techniques for preservation during the sectioning and with NMR, it's been shown that these pores are indeed unobstructive and allowing for mass flow to occur from one cell to the next. So we now know that these are the cell types that transport sugars longitudinally through a tube piping system in the plant. However, in gymnosperms, this is not clear. Here we're seeing two sieve cells adjacent to each other, one on the left and one on the right, in a pine tree or gymnosperm. And there are many, many smooth endoplasmic reticulum going through the pores, apparently occluding the pores. So it is not clear that sugars can transport so readily from one sieve cell to the next in gymnosperms. There's not been very much research done here, and at this point, there has not been any observations of unobstructed sieve pores in the sieve plate between two sieve cells in a gymnosperm, and more research is needed to clarify this issue. Such obstructed pores would result in high resistance to mass flow between the sieve cells, and so it's very unclear as to how sugar transport occurs in gymnosperms. P proteins can plug the sieve pores, and in this figure as well, you can see the formation of callose material. This is a beta-1,3 glucan similar to cellulose that is synthesized to help plug up the pores when a cell is damaged. So there are two ways of plugging the pores in a broken sieve cell or cut sieve cell. Those include the P proteins and the callose material. So we can summarize the characteristics of two types of sieve elements in seed plants. First, we have sieve tube elements found in the angiosperms, with some sieve areas that are differentiated into sieve plates, and individual sieve tube elements are joined together to form a sieve tube. These sieve plate pores are open channels to allow mass flow between cells. And there are P proteins, which are present in all dicots and many monocots. The companion cells associated with these sieve cells are sources of ATP for energy and perhaps other compounds. In some species, they serve as transfer cells or intermediary cells, which we will get to in a few moments. There are also sieve cells found in gymnosperms. At this point, there's no evidence that there are any sieve plates, but their sieve areas are very similar to those in angiosperms. The pores in these sieve areas appear blocked with membranes, and therefore it is unclear how mass flow could occur between these cells. There's no evidence of P protein in gymnosperms, and there are some cell types called albuminous cells that sometimes function as companion cells. So a very fundamental part of this lecture is that translocation in plants, and in particular the angiosperms, occurs by a mechanism known as source to sink. And what do we mean by that? The sources are sites of sugar production that produce excess photosynthase than what they need, such as mature leaves. Sinks are sites of sugar utilization. That is, they are either non-photosynthetic tissues, such as roots or trunks or fruits, or they are young, growing tissues that have expanding cells with immature leaves that are not producing enough photosynthate to export sugar at this point in time. Usually that time occurs at about a 50% maturity size of a leaf. And at that point, it starts to transition from a sink to a source. It's been very difficult to study the composition of the phloem sap. Most plants, because of the P proteins in callose tissue, block the sieve sap flow, and it's very difficult to collect anything. However, there are some plants that can be coaxed into leaking their phloem sap by a variety of techniques. Some will just flow a little bit anyhow. Others, we can use a material called EDTA, which binds up the calcium and prevents the callose formation. And there's another method by using aphids, as I'll get to in a moment. Suffice it to say that it's been a difficult problem to identify, but a number of people have been successful at identifying what the components of the sap are. Now, the main component of the sap is water, obviously. The next main component is sugar, and sugar is quite concentrated in the phloem sap. and these are in milligrams per milliliters of concentrations, and we can find that sugars are in the 80 to 106, and most of the sugar is sucrose, as we'll get to in a moment. The next major component are amino acids. Amino acids are transported in the phloem as a form of nitrogen transport. Nitrate cannot be readily transported through the phloem. So in most plants, it is transported as amino acids through the phloem. Amino acids are in the concentration in the phloem sap of about 5.2 milligrams per milliliter. Only 5% of the total amount of sugars that are being transported in the phloem. Next are organic acids at 2 milligrams per milliliter, proteins at about the same concentration, potassium between 2 and 4 milligrams per milliliter, chloride and phosphate and magnesium are even lower. There are other substances such as hormones and RNA, or particular messenger RNA, that can be also transported in the phloem and are used in signaling in the plant, as we will discuss later. So sugars are transported in the phloem as non-reducing sugars. These are sugars like sucrose that are not very reactive to things. Hexoses like glucose and fructose are highly reactive as they have an exposed aldehyde or ketone group, and this makes them not suitable for transport over long distances. There are other compounds or sugars that are transported in the phloem as sucrose bound with galactose molecules. These are compounds known as raffinose, which consists of a sucrose molecule and a galactose molecule, or stachyose, which is consisting of a sucrose and 2 galactose molecules, and even verbascose, which is a sucrose molecule with 3 galactose molecules. And there are sugar alcohols, mannitol and sorbitol, that are transported in some plants as well. These are plants that specialize in these particular kinds of transport molecules, but most plants are transporting sugars as sucrose. As I mentioned, nitrogen is primarily transported as amino acids, and the specific amino acids are actually the case, especially glutamate and aspartate, and their respective amides, the glutamine and asparagine. So associated with the Sieve elements are companion cells, and there are 3 types of companion cells. In some plant species, there's only one type of these companion cells. In other species, there might be 1 or 2 or even all 3 of these cell types. So let's discuss each of them. So there's the ordinary companion cell, there's the intermediary companion cell, and there are the transfer cells. These cell types are very important in the kind of phloem loading that occurs in the phloem sieve tubes, as we'll find out, and is responsible for causing transport through the phloem. We'll get to the mechanism of that towards the end of the lecture. But here in this figure, we can see 3 sieve elements in the center and 2 intermediary cells on the left and the right, and an ordinary companion cell down below the 3 sieve elements. So ordinary companion cells have chloroplasts with well-developed thylakoids. These are membranes within the chloroplasts that are important for photosynthesis. And they have a cell wall with a smooth inner face. The number of plasmodesmata in ordinary companion cells is quite variable and apparently reflects the pathway taken by sugars as they move from the mesophyll that are producing the photosynthate to the minor veins in the leaf. Intermediary cells have numerous plasmodesmata connecting them to the bundle sheath cells, as we can see in this slide and the subsequent slide after this. Intermediary cells also have numerous small vacuoles as well as poorly developed thylakoids and the lack of starch grains in their chloroplasts. So in this figure, again, you can see the 3 sieve elements with an adjacent intermediary cell, and you can see where the arrows are pointing to plasmodesmatal connections with the surrounding bundle sheath. This is for important transport processes that go on between the bundle sheath and the intermediary cells. Here we can see a transfer cell. Transfer cells, as we discussed earlier in xylem parenchyma, also exist in the phloem, and these are similar to the ordinary companion cells except for the development of these finger-like wall ingrowths, as seen here in this figure. As in the xylem parenchyma cells, these ingrowths greatly increase the surface area of the plasma membrane and increase the potential for solute transfer across the membrane. In these transfer cells, we can see relatively few plasmodesmata, yet the connections here are primarily between the transfer cell and the sieve element cell, as shown here. It is unclear what function these plasmodesmata serve between the sieve element and the transfer cell, but clearly they are important as it costs a lot of energy for the plant to make these plasmodesmata. They must be serving some sort of function in communication or transfer of components.
So I mentioned that sugar is transported through the phloem by mass flow. And in fact, those measurements have been made on phloem transport and have been estimated to be in the range of about 1 meter per hour. Now that's much, much faster than the rate of diffusion. As you recall, in an earlier video, we described the diffusion of water across a 1 meter long branch would take about 32 years. So clearly this is much, much faster than any kind of rate of diffusion. At the end of this video, we'll talk about the mechanisms for phloem transport as proposed by Munch in 1930, the pressure flow hypothesis. But in the meantime, let's look at some of the other mechanical aspects of this phloem transport, starting with phloem loading.
So phloem loading involves the transport of photosynthate molecules, those molecules that are produced by photosynthesis, and we haven't talked about photosynthesis yet, but suffice it to say that sugar is produced in the leaves, and then that is transported to the veins where it is loaded into the sieve cells.
Note that the sieve cell and the companion cell are known as the sieve cell companion cell complex. Phloem loading can occur through principally two different pathways, a symplastic pathway and an apoplastic pathway. Now, if you recall from previous lectures, I defined the symplast and apoplast in this way. That is that the symplast is everything inside the plasma membrane, and the apoplast is everything outside the plasma membrane of the cell.
Symplastic transport occurs inside the plasma membrane, inside the cell from one cell to the next, whereas apoplastic transport occurs outside of the plasma membrane in the cell wall space. So in this case, we're talking about phloem loading, that is the transport of sugars symplastically into the sieve cells or apoplastically into the sieve cells across the plasma membrane. So let's start first with symplastic loading here on the left. And we have a picture of a cross-section of a leaf with a vascular bundle, and we can see a number of mesophyll cells that are lined up transporting it to the center of that vascular bundle. And we see CO2 here in a mesophyll cell being converted into sugar. And that sugar is being transported symplastically through plasmodesmatal connections from one cell to the next, and eventually reaching the bundle sheath cell. And this, in turn, still has a plasmodesmatal connection that's connected directly to the sieve element through plasmodesmatal connections as well. That bundle sheath cell then is connected to an intermediary cell with plasmodesmatal connections, and ultimately that intermediary cell is connected to a sieve element. So in this case, sugar is produced inside the mesophyll cell and transported symplastically through plasmodesmatal connections all the way to the sieve element. In other plants, which is representing probably the majority of plants, there is a process called apoplastic loading, where the mesophyll cells, again, are producing the sugars by photosynthesis, and it's transported from one mesophyll cell to the next until it reaches the bundle sheath cells. And there it's transported into the phloem parenchyma cell by plasmodesmatal connections or to an ordinary companion cell. Note that the ordinary companion cell here has no plasmodesmatal connections. This forces the sugars to be unloaded out of either the bundle sheath cells or the phloem parenchyma cells, and then it accumulates in the apoplast. And then by a mechanism we'll describe in the next slide, there is an active loading process of sugars from that apoplastic region outside the ordinary companion cell into it. And then through plasmodesmatal connections between the ordinary companion cell and the sieve elements, the sugars are transported into the sieve element. So we see in this slide here that sieve element companion cell complex where there are no plasmodesmatal connections between the parenchyma cell or bundle sheath and the companion cell. And so in this plasma membrane of the companion cell, we have a mechanism for active transport of the sugars into the companion cell. And this is key to driving transport by mass flow through the plant cells in this kind of apoplastic loading mechanism. And we know the mechanism here. There is a proton pump that we talked about before when we talked about ion transport that pumps protons into the apoplast from the cytoplasm using ATP. So it's using energy and it's electrogenically pumping protons actively against its electrochemical potential gradient. And those protons then are used as an energy source to drive sucrose transport into the cytoplasm of the companion cell from the apoplast. Again, the sucrose concentrations are actually much higher inside the cell than outside the cell, but it's using the energy of the protons to go down passively down its electrochemical potential gradient now from high proton to low proton concentrations and drive sucrose transport through a symporter, a sucrose symporter that uses protons and sucrose. Cool, huh? And in this way, a concentration of sucrose can be built up inside the companion cell at a much higher concentration than what is in the apoplast, using the energy from the protons, which ultimately came from the ATPase that was driving protons into the apoplast. Can't forget any of those earlier videos and lectures that I gave, that this is all connected. In order to understand the whole picture, you've got to understand each of these components and parts. And another mechanism of phloem loading using intermediary cells, there's another system that has been documented that involves the synthesis of sucrose, which occurs in the mesophyll cells. And that sucrose is then transported into the intermediary cells, where the sucrose is utilized with galactanol to produce raffinose. And raffinose is then transported into the sieve element. Turns out that raffinose is a larger molecule and cannot diffuse back into the bundle sheath cells. So by utilizing the sucrose to form raffinose, it decreases the concentration of sucrose in those intermediary cells. And this keeps the flow of sucrose from the mesophyll and bundle sheath cells into the intermediary cells. The raffinose is then transported into the sieve elements where the sugars build up in the cells and are then transported through the transport pathway of the sieve tubes. Again, this is a less common method for sugar transport in plants, but it is one that has been documented. So let's look at some of the patterns here for apoplastic and symplastic loading. If we look at the features here for apoplastic loading, the transport sugar is sucrose. And in the symplastic polymer trapping method, we have raffinose or stachyose in addition to the sucrose being transported through the phloem. There's also another mechanism that we first referred to, which is this passive symplastic loading of sucrose by plasmodesmatal connections. And again, this is sugar or sucrose transport. The cells that are involved in apoplastic loading are the ordinary companion cells and or transfer cells. In the symplastic polymer trapping using raffinose and stachyose, intermediary cells are involved. And in the passive symplastic loading, we have ordinary companion cells. Another striking feature that's different between these is the number and conductivity of the plasmodesmata connecting the Sieve element companion cell complex to the surrounding cells. So in the apoplastic loading, there is either no plasmodesmatal connections or very few. In the symplastic polymer trapping, there are a high number of plasmodesmatal connections, as well in the passive symplastic loading mechanisms. In addition, in our apoplastic loading mechanism, it's transporter driven by the proton sucrose transporter and proton pumps. In the symplastic polymer trapping, we're independent of any transporters, as well as in this passive symplastic loading mechanism. Overall, the concentrations of transport sugars in the source leaves, the ones producing the sugars, are low in both the apoplastic loading and symplastic polymer trapping. But the passive symplastic loading requires high amounts of sugar being produced in the source leaves. And again, the cell types in which the driving force for long distance transport is generated is the Sieve element companion cell complex for apoplastic loading. It's mainly herbaceous plants that occur. In the symplastic polymer trapping, it's the intermediary cells, which are largely in herbs and woody species. And in the passive symplastic loading, it's through the mesophyll, and it's mainly trees.
So a word about phloem loading and phloem unloading. People have tried to increase plant production by increasing the source, that is, increasing the rate of photosynthesis in leaves. But in fact, as we'll find out later on, growth is largely regulated by the sinks. And sinks are dependent upon the sink strength. And sink strength is dependent upon the size of the sinks and the activity of the sinks. So although photosynthesis can increase temporarily when CO2 concentrations are increased or light intensities are increased or other factors, ultimately, photosynthesis has feedback regulation and is downregulated when there's not a demand for the sugars. This makes sense, actually, when you find out that rubisco, which is the key enzyme for binding CO2 and fixing it into sugars, is 50% of the protein of a plant. And so it's a very expensive process, photosynthesis. And therefore, if there's no demand for it, there's no need to upregulate it the way it can be.
So it's demand driven. And it is the amount of growing materials and sinks that drive photosynthesis. So photosynthesis will increase when there's higher sink demand, and photosynthesis, as I mentioned, will decline when sink demand declines. And there's been a large number of experiments to show that this is actually happening. So the important driving pathways, in fact, are the phloem unloading, which we'll look at here. So in the top figure here, we can see that phloem unloading can occur symplastically and over short distance transport pathways. So if we go to the sink, which might be a root or a fruit or a growing leaf even, or a trunk, the companion cell Sieve element complex in those organs will have direct plasmodesmatal connections with the adjacent cells. And you can have one layer after another layer after another layer going all the way out to the epidermal cells of that organ where symplastic transport will occur. In addition, there's an apoplastic pathway for phloem unloading where there can be a block in the symplastic transport pathway which requires a unloading of the sugar into the apoplast and an active transport in which that cell actively takes up sugars in a similar mechanism to phloem loading with a sucrose transporter. So in number 1 here, we see the sieve element complex with the companion cells being isolated symplastically. So there are no plasmodesmatal connections between that and the adjacent cell. In example 2a, we have an additional layer. In example 2b, we have additional layers, and it's not until we get to the last sink cell or epidermal cell that we have this phloem unloading step occurring. So it just varies depending on the organ, the tissue, and the plant species as to which of these pathways occur. So let's summarize phloem transport here for a moment. Sugars are transported through sieve tubes. What I didn't mention before is that transport can occur bidirectionally. That is, we can have transport of sugars moving up the plant and down the plant. Unlike in the xylem, where it is largely unidirectional from the roots to the leaves, sugars can move from the leaves to a growing tip, or down to fruit, or down all the way to the roots. We'll understand this mechanism of how this happens in a moment, but suffices to say that a key factor here is that it's not occurring in the same sieve tube, but in separate sieve tubes in the same vascular bundle. So we can have flow going down one tube and up another tube, but not happening in the same tube going up and down in the same tube at the same time. The direction is always source to sink, and we'll understand this better when we talk about the pressure flow hypothesis. And the source, again, are the photosynthetic leaves that are exporting sugars from the leaves. That is, they are producing more sugars than they are utilizing. And the sinks are non-photosynthetic tissues and organs that are growing or not photosynthesizing at all, or storing sugar as in the form of starch. So that brings us to the Munch pressure flow hypothesis. So to understand this mechanism, we have to understand water transport and ion transport and mass flow. So all those lectures before where we talked about these things in detail were important for understanding how this mechanism works. And Munch, as I mentioned, proposed this pressure flow hypothesis in 1930, and he actually constructed tubes and glass vessels to show that this system could work in a mechanical way, not even in a living tissue, but the components that he built all exist in living tissues within the plant and will be explained here. So flow through the sieve tubes is a passive process in response to a pressure gradient. It is the pressure gradient that is driving mass flow. And this is the most important thing to understand, that we're going to separate out the osmotic processes that are occurring here from the mass flow processes. They operate under two different principles. So water is moving by mass or bulk flow through these sieve tubes, driven by the pressure gradient. The key here is because there are no membranes between the cells, between the sieve elements that form the sieve tubes, the osmotic component in the phloem has no effect on water movement, and only the pressure gradient becomes important for water movement. However, water does move by osmosis into the cells across the membrane of the companion cells and the sieve elements. They do have plasma membranes with adjacent parenchyma cells and xylem cells and transfer cells that can allow water movement across their plasma membranes and into the sieve cell companion cell complex. So what's the evidence for the pressure flow hypothesis? First of all, we can say that the phloem anatomy pathway exists. That is, that we have cell types that are able to fit the descriptions that are necessary with plasma membranes or the lack of plasma membranes. And we'll show a model here at the end to show this. We also know that sucrose moves by mass flow and not diffusion. That is, it moves at much, much higher rates than diffusion. And we know that a pressure gradient exists. One of the clear evidences for this are the fact that aphids can bite into the phloem and exude sap, as I'll show in a moment. But we have other evidences where people have attempted to measure with pressure probes the sap in the phloem and have determined that it is under pressure. However, the best evidence, in my opinion, is that of aphids. So aphids bite into a plant tissue, and their stylet is able to find a sieve element. So here we can see on the right-hand side an aphid stylet actually puncturing into the sieve element, specifically targeting the phloem sugars. And it seems that the aphids have some sort of protein or chemical that keeps the sap flowing. And in this picture on the left, you can see the exudate coming out of the anus of the aphid, causing what we call often honeydew. So scientists have gotten very clever, and this is in fact how they discovered or identified what the sieve tube composition was. Is they anesthetized aphids with, I think, CO2, and then they zapped them with a laser so that the body was no longer there, but the stylet was still punctured into the sieve element. And because the sieve tube element's under pressure, it forced the sap out the end of the stylet, and the sap could be collected. Now, it's a very small amount of sap coming out of one of these, so they had to put in quite a few stylets and collect it over a large period of time to collect enough sap to analyze it. But this is clear evidence that the phloem is under pressure. Now, there have been other hypotheses in the past that had suggested that there were other mechanisms for phloem transport. I will not go into them, but let's suffice it to say that the Munch pressure flow hypothesis is a generally accepted mechanism by which phloem transport occurs and is largely substantiated by a large body of evidence to support that. And this is a passive process, not an active process. The sugars are moving by mass flow down its pressure gradient. Okay, so let's look at a model to see how this occurs. So in this model, on the left-hand side, we have some xylem vessel elements forming the xylem and let's say a stem. And on the right-hand side, we have the phloem sieve elements forming a phloem tube system for movement of sugars. And in between, there might be some adjacent parenchyma cells like the xylem parenchyma or the phloem parenchyma, in which transfer of water can occur. So in this mechanism, we can look at the xylem here, and we can see that the xylem, as we've learned in the past, operates in a certain mechanism under tension with a measurement of water potentials that are in the negative values. And if we look at the lower cell in the xylem, we can see that the water potential is equal to minus 0.6 megapascals. And the pressure potential is, as psi p here, is equal to minus 0.5 megapascals. That is, it's under tension. And the solute potential, there are some solutes in the xylem that create a solute potential of negative 0.1 megapascal. Now, if you don't remember what the meanings of these things are, then I highly suggest you go back to my earlier videos on water and the definition of water transport and how the chemical potential works. So this water will move up through the xylem, and in this particular example, they're using a water potential of minus 0.8 megapascal. So it's lower. So water would naturally move this way if there were some membranes, but that's not relevant in this particular case. What's relevant is that there are adjacent cells that have plasma membranes, and for the water to move from the xylem into those adjacent cells, it has to cross a membrane, and osmosis comes into play. So if we look at the water movement from the cell that has a minus 0.8 megapascals with a lower tension now, because in fact there is a lower tension in the upper cells of the xylem because it's closer to the leaves, which is creating that tension by evaporation or transpiration. Now, as we focus on the phloem here on the right at the top, we have some companion cells and sieve cells, which are surrounded by plasma membranes. And there's a process of loading here, which is shown here on the right from the source cell as an apoplastic loading. These red spheres represent sugars. They're being actively loaded into a companion cell, and that companion cell then is transporting those sugars into the nearby and adjacent sieve elements, and those sugars are then becoming more concentrated because they're getting loaded into there. So in this particular example, we have a water potential inside the phloem cells at minus 1.1 megapascal. Now, this is not important for pressure, but it is important for water movement across a membrane, which is coming from the xylem or the adjacent cells in the phloem parenchyma. And water is moving from what's shown here in the xylem at a higher water potential at minus 0.8 megapascals down its water potential gradient to a water potential of minus 1.1 megapascals. So this is water is moving passively in response to the much lower solute potential, because the solute potential is minus 1.7 megapascals as compared to the xylem, which has only a minus 0.1 megapascal. This also generates turgor pressure because we now have water movement going into the cell driven by the solute potential that increases the turgor pressure to 0.6 megapascals. Following me so far? If not, go back, review what I just said, because the next point is that now we have built up a pressure gradient in that plant where the sugars are being loaded into the phloem. And it's moving passively by pressure through the sieve tube down a pressure gradient, because at the lower end near the sink, we have sugars being removed out of the phloem into the adjacent cells and finally into the sink cell. This results in sugars being unloaded, and this increases the solute potential inside the phloem so that it now has a sugar concentration much lower than up above, and we only have minus 0.7 megapascals, which causes the pressure in that cell to drop to 0.3 megapascals. So we're going from a gradient of 0.6 megapascals up above down to a pressure gradient below. This is what's driving movement through the phloem, that pressure gradient. However, besides the sugars leaving the sieve tubes, water is also leaving the sieve tubes. Where does it go? It goes back over to the adjacent xylem. How so? Because we can follow the water potential gradient. Remember now that that cell back in the sieve tube is minus 0.4 megapascals. But the water potential in the adjacent xylem is lower at minus 0.6 megapascals. And as a result, water passively flows down its water potential gradient into the xylem. So here we can see a loop, a cycle of how the xylem and the phloem are working together using both osmosis and mass flow. The osmosis is important for driving water into and out of the phloem, And it is the pressure gradient forming in the sieve tube that is driving phloem transport from a source to a sink. And this is how source to sink is driven. Competition can occur between sources and multiple sinks. And we have a definition here then for allocation of sugars versus partitioning of sugars. So allocation is the process of regulation of the photosynthate that is produced into different metabolic pathways within the cell. For example, the sugar that's produced in the cell could go into other export pathways for sugar to other cells, or it could go into the formation of starch where it might be stored in the chloroplast, for example. We'll get into this more when we talk about photosynthesis. But there's another component known as partitioning, which is where there's competition between sinks in a plant. And this is quite a common, if not universal, process that's going on in a plant. So on the right-hand side here, we see a small Arabidopsis plant that has been specifically designed with a Gus marker. Now, I'm not going to get into the biochemistry of this, but suffice it to say that where you see the blue staining is the result of a sucrose transporter, a sucrose proton symporter that we talked about for phloem loading, that is being expressed in the leaf tissues. And what you can see in the center is the young leaves are completely white. There's no blue showing up. So the sucrose transporter that is responsible for phloem loading is not present in those leaves. That is, those leaves are sinks, not sources. They are not exporting sugar. And then as we get to older leaves, we begin to see the development. So on the left-hand bottom corner, you can see the blue starting to form at the tip of the leaf. And this is where the sucrose in the leaf is first being exported as that leaf begins to mature. And then in the more developed leaves, you can see that this is going throughout. And notice that the transporter is expressed right along the veins of the leaf. So again, it's showing its role in phloem loading. Now there's another kind of competition, which is shown here on the right-hand side, which is where we have different sinks. And here we're seeing the phloem as transport with the blue lines coming from source leaves. And we can see that those leaves are transporting sugar up the plant to sinks that are above the leaves, in this case the shoot tip, and also the fruit, in this case a tomato plant, and down the leaves to the roots. Notice that the transport pathways for up and down are on different lines, indicating they're going through different sieve tubes. Now the sink for the fruit, the tomato, is competing with the sink for the shoot tip. And you could argue that the fruit is closer to the source and therefore gets first dibs on exporting the sugar. But it's also driven by the strength of the sink. And we define sink strength as the sink size and its activity. So if there's a high unloading or high demand, as there might be with fruit, and there usually are, fruit are some of the strongest sinks available, they will divert the sugars away from the shoot tip. And the shoot tip then will no longer get the sugars that it needs as much, and it may slow its growth, or it may stop its growth altogether. Likewise, the roots are also competing for those sugar sources. So how is the plant deciding which ones? Well, there may be multiple mechanisms for this, but one of the primary ones is going to be that pressure gradient. We're going to see stronger pressure gradients driving sugar in that direction. So the more unloading that goes on, the lower the pressure, the stronger the pressure gradient, the more sugar will flow in that direction. It's as simple as that. So in a tomato plant or a grapevine, which I like to grow, there's competition going on between the roots, the fruit, the shoot tips, the trunk. Everybody's trying to get their fair share of the sugar. And in some way, the plant balances that out. At times in the season, you may see more stimulation of root growth, for example, in the early spring. And as the season develops over time, the shoot tips then will start to develop and produce more leaves, to produce more sugars. And then the fruit will develop, and the fruit will act as the major sinks and drive photosynthesis, but drive sugar transport at the sacrifice to the other sinks. Agricultural scientists call the harvestable part of the plant, or the proportion of the harvestable part of the plant, as the harvest index. And they use this understanding to manipulate and breed for plants that have a better harvestable index. So they get more food per plant. And here you can see what happened during the Green Revolution, where plant breeders bred for dwarf wheat, which meant that they had lower amounts of stem and lower amounts of leaves, but higher amounts of grain proportionally. And as a result, increased yields dramatically by as much as 10 times the amount of yield. This is known as the Green Revolution. It eliminated famines in many parts of the world. In fact, India has not had a famine since 1943 as a result of these impressive gains in crop production. Through the understanding of knowing how crops are partitioning food to different aspects of the plant, you can design plants to be more optimal. When I was a graduate student at UC Davis in the veg crops department, breeders had been selecting for tomato plants with higher and higher yields by increasing the amount of fruit that the tomato plant would produce. But in fact, they got the plants out of balance, and as a result of that, the fruit didn't develop and fully ripen properly, and the roots could not supply the tomato plants with enough potassium, and they started to suffer potassium deficiency in the leaves. So there's balance in a plant that is required, and we can manipulate and adjust them to optimize that balance, but balance is required for any plant. In summary, phloem transport of sugars is driven by a pressure gradient generated by phloem loading and phloem unloading from the vascular tissues. Phloem transport occurs by mass flow from source to sink. And knowledge of source-to-sink relationships and their regulatory controls is leading plant breeders to produce plants with higher efficiencies and crop productivities.
Well, that's it. I hope you enjoyed this video. If you like this video, then please like it on my YouTube channel, as there are going to be more lectures on plant physiology. The lectures will become more biochemical in that we will be covering photosynthesis, respiration, starch synthesis, and then we'll take all that information and we'll integrate it into how plants grow. We need to know all these factors to better understand the plant growth of plants. Then we'll go into hormone regulation and how they act as plant growth regulators. And finally, we'll cover how environmental stress impacts plants and how they acclimate or adapt to these stressful conditions. Have a great day.